In this video, I'm going to be talking about how cells divide, which is a process called mitosis. So here's a picture that I thought was kind of cool showing an onion root tip in a regular lab. You would normally get to see this, but you can see the different stages of mitosis happening here, and we'll talk about those as we move along. So a good question is, why do cells divide at all? Why do they not just get bigger? And that's because for normal cell development, your body needs to be able to regulate when things are dividing and how much they're dividing. It really needs to have a control on that because when cells grow out of control, you get something like cancer. So there's also a level of difficulty moving nutrients and waste in and out of a body the larger that it gets. It also puts more demands on the DNA. This is a good slide that basically is saying a lot of the limits that our DNA has and why it can't get um, control a cell that is much bigger than what it is. And then this is a good slide talking about how if you have something like the nucleus, if it's surrounded by something small, it's easy to get waste in and out and to communicate with something small. But if that same nucleus is in something much larger, it's a lot harder to get waste in and out, to get messages in and out. And remember that a lot of these processes happen with just simple diffusion, with the waste going out and the nutrients coming in, water coming in and out. So when you have something that's trying to come all the way from the center of a very large space, it's really hard for things to diffuse in a timely manner. So, our replication by mitosis is somewhat complicated. We'll go over it, but first I wanted to show you the binary fission, which is how prokaryotes, which are bacteria, how they divide, which is a pretty simple process. And this video kind of walks you through that.
To understand binary fission, let's look at a simplified version of our bacteria to better show the bits that you need to understand. Bacteria have both a large circular piece of DNA, which contains the important genes that they need for basic survival, plus extra smaller circles of DNA called plasmids. When the bacteria wants to divide, the cell firstly replicates all of its genetic material. The cell then grows, and the two large circular strands move to opposite poles of the cell. However, the smaller plasmids are more randomly distributed. Finally, the cytoplasm itself begins to divide, and a new cell membrane and cell wall form around the new cells. Here, we've shown the wall and membrane as a single line. But don't forget that in reality, there are two layers: the cell wall on the outside and the cell membrane on the inside. As binary fission is such a simple process, prokaryotes can divide very rapidly, given the right conditions, which generally means a warm environment and plenty of nutrients. For example, some bacteria can divide every 20 minutes.
Okay, so binary fission, it's how prokaryotes are splitting. And like it said, the crazy thing is their division process is very simple, but then also um, what they need is like basic nutrients and a certain temperature that they're used to, like a range of temperature, and they can divide some of them up, until every 20 minutes. That's really quickly. And if you think about like the doubling of viruses or even bacteria and how they get doubled, this is what they call exponential growth, where it can just happen so rapidly, especially if it has a shorter replication time, like in binary fission. So cell division happens so that we can grow and also so that we can replace damaged cells. So cell division happens and it results in two daughter cells from one mother or parent cell. So this one larger cell makes copies and divides and forms two identical daughter cells that are identical to the mother. They don't have half of the DNA because this was replicated before the division. So they actually are identical. At first, they are smaller, but they immediately begin growing to their normal cell size. Inside of each of these cells, you have about 2 meters of DNA. If you think about that, 2 meters, a meter is like floor to your doorknob. Um, picture 2 of those meters inside of every cell of your body of DNA. It's so much. I've seen, things you can look at. If you put your DNA end to end, that it can reach so far in space. It's just interesting that that's all curled up inside. And normally you can't see it. But each cell does get a complete set of the DNA in the end, and each species has a certain number of chromosomes in them. So humans have um, 46 total chromosomes, and they're in pairs, so it's like 23 pairs of chromosomes. Now, in our sex cells, they're not in pairs because the egg gets paired with the sperm. So, specifically, just in our sex cells, there's 23 individuals, but we'll talk about that later on. So, um, during eukaryotic division, each cell first makes a copy of its chromosome, and then each daughter cell gets one of those copies. And there's two main stages. The first stage is mitosis, which is the division of the nucleus. And then after mitosis, there's another process called cytokinesis. And cytokinesis is where the cytoplasm is divided out, and so are all of the organelles like mitochondria, lysosomes, Golgi, ribosomes. They're all just like sorted out and divided between the two new cells. So chromosomes are generally not visible at all except during division. And what happens is these coil up, they get really coiled up, and then and condense and basically make these really cool chromosomes. And they have two identical copies, and they're called sister chromatids. They're held together at the center by something called a centromere. And so these are two identical sister chromatids that are held together by a centromere. So the cell cycle is this: the majority of the cell cycle, what's called the cell cycle that's going around and around, G1, S, and G2 phases are all something called interphase, and it's a really long time period. It's basically all of the preparations that are being made. The cell grows, it prepares for division, and it will eventually, as part of mitosis, divide and make the new daughter cells. So here's the major phases: the G1, the S, the G2, mitosis, and then cytokinesis. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm and all the cell parts. So, in the interphase, here is what is happening. In this big long part of this, from here to here, is called interphase. And a lot of work is happening in interphase. It's not just hanging out. The cell is doubling in size. There's enzyme, cytoplasm, everything's getting doubled. And then here, there is a checkpoint. If the cell has not replicated properly, either the problems will be fixed or the cell will actually lyse and basically die. So, are the options here. So, it has this checkpoint to make sure that things are going well. In the S phase, there's replication of the DNA. And if there's a replication problem, you can blame it on the S phase of the DNA. If there's a replication problem of the DNA, then that was because of the S phase. For example, if there's only one copy of the gene, then the mistake happened in the S phase. In the G phase, I apologize, in the G phase, this is where special structures that are only used during cell division are made and assembled. You can think of like the spindle fibers and centrioles that will end up pulling the cell apart, and those are made in the G2 phase. So in interphase, which this part would be the G2 phase, you're going to get the centrioles made. They're part of the special things that are only used here. And interphase also accounts for 90% of the cell's life. So this interphase, 90% of the cell's total life. Um, and then there's centrioles here. These centrioles, I want to talk about them a little bit more. They will actually grow spindles out of them, which are microtubules, which will pull the chromosomes into place and actually pull the chromosomes apart. So they're really important pieces to this whole process. So in interphase, the cell also doubles in size. The chromosomes have duplicated. Again, that was in the S phase. And then the cell makes these special structures that are going to be needed for the division process of mitosis. So at interphase has G1, S, and G2 phase. And it's all the time before mitosis. So it's that long piece of the pie, those three big pieces of the pie. And then mitosis is broken down into 4 stages: prophase, metaphase, anaphase, and telophase. And so I like to go back and look at this picture because it shows different, this is just an onion root tip cells. And if you look at it under the microscope, you can actually see the cells going through this process, which is pretty cool. So the first phase of mitosis is interphase, prophase. And in prophase, the chromosomes start to coil up. The nucleolus, that is what is surrounding the nucleus, it will disappear. Each half of the chromosomes double up, and they are bound together by a centromere. And the centrioles start to move. These are the guys that are going to rip the chromosomes apart once they're all nicely coiled up. They move to opposite sides of the cell and start to grow spindles, which are basically microtubules that are just going to rip the chromosomes apart. Then you move on to late prophase, where the nuclear membrane forms. The spindles start to attach to these chromosomes, and they're going to start attaching so they can pull them into place where they need to go so they can separate them. The next phase is metaphase, where these microtubule spindles, led by the centrioles, have pulled these chromosomes along the equator, and they have them lined up here. In metaphase, I remember they met in the middle for metaphase so that they can get pulled apart. And because these are sister chromatids, remember that anaphase is where two sisters are like yelling to each other like Anna, Anna, because they're getting ripped apart. So these chromosome halves are getting ripped apart. The centromeres, which were holding them together, have now been ripped apart by these microtubules called the spindle fibers. Led by the centrioles. So they're being ripped apart, and the chromosomes are getting pulled to the opposite side. The good thing about this is that now each side is going to have an identical copy of the chromosomes. The next part is the nuclear membrane starts to grow, and then you get something called a cleavage furrow. The cleavage furrow is formed by proteins, which are called the FTSZ proteins. But it basically causes this pinching of the middle of the cell to result in two cells that are identical, exact copies of each other. So here are different phases in the process. You can see a bunch of these are in prophase, and here's one that's in prophase. Here they're getting torn apart like Anna, Anna. They're yelling to their sisters as they're ripped to opposite sides. Here's another one of those. And then you can see the telophase where it's just newly split. Um, so just some things to look at there. Okay, once the telophase has happened, then you get to cytokinesis. And remember, cytokinesis is the division of the cytoplasm, and it's also the division of all the organelles within the cell. If you looked under a microscope and you saw a cell that looked like the number 8, where it was just about two separate cells, then it would be undergoing cytokinesis. This usually happens at the same time as telophase. So these are almost happening simultaneously, but where telophase is really the dividing piece of it, um, the cytokinesis is more the division of the goods, whatever is there, the cytoplasm and the organelles. It's really the division of the different parts inside of the cell. So here's a picture of cytokinesis in the animal cell and the cleavage furrow that's there. Remember, this cleavage furrow is formed by the FTS Z proteins. So cytokinesis in plants can't really pinch in the middle because they have that really rigid hard cell wall that surrounds them. So instead a plate, and you can see a baby little plate starting to form here, basically a plate will form inside of plant cells, and that plate will cause the division. So if you were to look at people's chromosomes under a microscope, it's actually really cool to see these. So here you can see when you first get all the chromosomes, they will be not paired up and ordered like this. So a lot of times you have to make pairs. So this is something that we do at research stations. I've done this like with condor DNA a number of times, where you can take images of their DNA and match it into its pair. And then you can see if there's any problems or anything that's different. And you can kind of see how each of these are paired all along the way. Except if you look at this one, can you see how this side is the left is a lot bigger than the right? That's because this is the final pair in a human where it has an X, but then it has a Y. So it has an X and a Y chromosome that are different, meaning that this is a male. If you looked at this karyotype of DNA, you can go along here and they're all matched up. And then we get all the way to the 23rd pair, and there's no Y. There's no Y. Both the X's match. There's two X's and they match. So this is going to be a female. Then you can also go along on some karyotypes and find something like this, where there's 3 right there. Or you can look at the fact that there's a Y missing. Well, first of all, let's diagnose the gender of this individual. If there's 2 X's and no Y, then this is a female. But here we have 3 chromosomes on the 21st pair. This person has something called trisomy 21 or Down syndrome. Trisomy, 3 sets of genes at 21. So they're going to have Down syndrome because every cell of their body will have 3 copies in the 21st pair. I actually have a nephew with Down syndrome, so something that I think, I don't know, I love it. I love him. He's a great nephew. Okay, so then here on this karyotype, I wanted to point out the end of the arrow. So here's a tiny little arrow and it's pointed here, where this chromosome is just a little bit shorter. And then here on the 23rd pair, you can see a longer chromosome and a shorter chromosome in the Y. So you can see there's an X and a Y. And the X and the Y, it's normal for them to look different and be different sizes. The Y is always considerably smaller than the X. So the X and the Y chromosome, this is going to be a male because there's an X and a Y. But having number 11 be a little bit shorter, this individual has something called Jacobson syndrome. And it's basically the shortening of number 11. The terminal end of this is actually it's actually been deleted, and so that's why it's shorter. So you could discover this by doing a karyotype of the white blood cells on the individual. So Jacobson syndrome can cause heart defects, sometimes intellectual deficiency, sometimes bleeding disorders, but sometimes your doctor will do a karyotype just to see if there's any abnormalities in the karyotypes of the genes. So, to recap mitosis, it takes a while. It takes around 30 minutes to 2 hours. One mother cell makes two daughter cells, and the two daughter cells are totally identical. The result is that for unicellular plants and animals, this is a type of asexual reproduction. So, mitosis is just the splitting of your cells. So, you didn't need sexual reproduction for that. It's an asexual process. And in multicellular organisms, it helps repair and it helps grow the organism. Oh, I'm sorry. And then the importance of mitosis is that you get exact duplicates and the daughter cells can carry out the same functions as the parent cell. So there's a comparison of mitosis, meiosis, which is in the sex cells. We will talk about that in the next chapter, and binary fission. Mitosis happens in almost all cells of the body, like your blood, your nerves, your skin. It just isn't happening in your eggs and sperm. So meiosis is happening in the eggs and sperm, and binary fission is happening in bacterial cells. So the regulation of the cell cycle, um, there are certain cells that divide all the time. For example, skin cells are dividing frequently, hair, nails, their blood is dividing often. But then there's other parts of your body like liver cells where liver cells are able to divide, but they rarely do. And then there's certain cells like muscle cells and nerve cells that tend to not divide at all. Muscles, when they want to grow bigger, for example, they just enlarge. Um, so then controls on cell division. When cells are growing and they kind of bump into each other, they stop growing. They go, oh, okay, that's my limit, and they'll generally stop growing. So when you get an injury, though, the cells on that outer edge will start growing really rapidly until you're healed, and then they will slow down. There's a lot of cell regulators in your body that make sure that this process stays in check. And most of these are all proteins. So there's proteins that regulate the cell division by speeding it up, slowing it down. They start or stop it, and they stop uncontrolled cell growth. So uncontrolled cell growth is like cancer. So cancers are just your regular body cells that are growing uncontrollably. And when they grow uncontrollably, they have, um, for example, something called proto-oncogenes. They have sometimes failed to do their job. Their job is to regulate cell growth. And if they have not, then sometimes those genes can be oncogenes where they are cancerous and they'll grow uncontrollably. Sometimes they break off, travel to other parts of your body. Um, and the problem there is that they grow uncontrollably in places they don't belong, using all kinds of nutrients. So, an example of a mutation that could cause cancer, if you have a protein that controlled the cell cycle that was mutated in a regular liver cell, then all of the future daughter cells from that mutated cells, all of the future daughter cells would also grow uncontrollably. And then all of their daughters would grow uncontrollably. So that's one way you could get liver started or a cancer started in a place like the liver. One last thing that is just pretty interesting, and you might be tested on this, so just know about horizontal gene transfer. Horizontal gene transfer is when some organisms actually steal genes from other organisms, which is just cool. So here's a quick little video to end on that.
What is horizontal gene transfer? Most of us are used to the idea of vertical gene transfer, that is, the passing down of genes vertically from parent to child. Hopefully, that's all pretty straightforward. In contrast, horizontal gene transfer, sometimes called lateral gene transfer or sideways gene transfer, is exactly what it sounds like. It is the passing of one or more genes through routes other than parent to offspring. Horizontal gene transfer is fairly common in bacteria. Some types of bacteria do this by actively attaching to and then sharing genes with other bacteria around them, even completely different species. Some bacteria can collect genes that have leaked out from other cells, use them as their own, and if all goes well, pass the foreign genes on to the next generation when they reproduce. Because of this, once a gene for antibiotic resistance evolves in one species of bacteria, it can quickly spread to others, which causes huge problems for us when trying to
Okay. So this is just an interesting process. Like horizontal gene transfer, for example, happens in sea slugs. They actually will steal chloroplasts from algae, and the chloroplasts basically, use the light energy to make energy. So sea slugs are not plants, and they don't normally have chloroplasts, but they will steal the chloroplast from algae and use those chloroplasts for their own photosynthesis. This is like amazing. Anyways, have a great day, and I hope that helps you understand chapter 6.